Assalamualaikum, and today Group 5 will explain about our research paper related to the next generation sequencing in human microbiota. This is from page of our article. The research paper that we choose is Insight into the Unique Lung Microbiota Profile of Pulmonary Tuberculosis Patient using metagenomics next generation sequencing. This article being published on 23rd of February 2022 by journal named Microbiology Spectrum. There are many authors that participate in making this paper, but all of them decide to let Guoliang Zhang and Liang Yang to be corresponding authors. Corresponding author is individual who is working on paper with multiple authors and manage to take responsibility for communicating with the journal that intend to publish in. Affiliation is a description of association with the organization or university. For corresponding authors, affiliation can be detected from article because it already stated that Guoliang Zhang, who was doing research at National Clinical Research Center for Infectious Disease at Shenzhen Third People Hospital, Southern University of Science and Technology, Shenzhen, China, and currently working under Guangdong Key Laboratory of Regional Immunity and Disease, Shenzhen University School of Medicine, Shenzhen, China, is one of the corresponding authors for this paper. Alongside Guoliang Zhang, Liang Yang also chosen to be the next corresponding author. Right now, he is working and doing research at School of Medicine, Southern University of Science and Technology, Shenzhen, China.
What is microbiota? Microbiota is usually defined as the assemblage of living microorganisms present in defined environment. Lung microbiota. The lung microbiota is a pulmonary microbiome community composed of a wide range of bacteria present in the lower respiratory system, mainly on the mucous layer and epithelial surface.
For tuberculosis is a bacterial infection that spread by the inhalation of tiny droplets from an infected person coughing and sneezing. Mycobacterium tuberculosis is the bacteria was being caused the tuberculosis. Metagenomic next generation sequencing. We will use metagenomic next generation sequencing (MNGS) to identify the lung microbiota profile of people with pulmonary tuberculosis in this study. MNGS is a shotgun sequencing approach in which all the nucleic acid DNA and RNA in a clinical sample is sequenced. The ability to sequence and identify the nucleic acids for metagenomic analysis can identify the genetic material from entirely different kingdoms of organisms. In this study, MAGS is applied to characterize lung microbiota by using bronchoalveolar lavage flood, bath sample, and throat swab. Throat swab was used for the healthy participants.
Based on the article, sequencing is used to characterize the lung microbiota using bath samples from selected patients that have TB background and samples from healthy patients. It helps to identify about the diversity of microbiota contained in the sample to give better information about TB pathogen.
This is the general step for metagenomic next generation sequencing. First, genomic DNA is extracted and fragmented. Second, adapters are attached for barcoding and library sequencing preparation. Third, the fragments of DNA are simultaneously and independently sequenced. Fourth, human-related DNA sequence reads are removed. 5. Cotics of long DNA stretches are assembled from shorter overlapping sequence. These cotics are aligned to a reference database for taxonomic classification.
First step in this technique requires to collect 5 different samples, which is sample from untreated pulmonary TB group, treated pulmonary TB group, cured pulmonary TB group, healthy group control, and lung cancer patient group. Next step is proceed with DNA extraction. The total DNA was extracted from both samples using Omega Biotech made by Universal Pathogen Kit.
The third step is metagenomic sequencing. The extracted DNA sample were used for metagenomic sequencing. 1 microgram of total DNA per sample was used as an input material for sequencing library construction. The library preparation was sequenced and pair and raw read was generated. The next step is data processing and sequence analysis. The raw read were filtered to remove adapter and low quality reads. The quality of processed read were assessed. The pre-processed read were mapped to the reference human genome and matching reads were removed.
Next step is taxonomic classification. After the reads were filtered, the remaining reads were taxonomically classified using the Kraken 2 software with the Kraken 2 bacterial and archaeal databases and default settings. Kraken 2 is the latest version of Kraken as it is very fast taxonomic classification system and has high accuracy in assigning taxonomy label using exact camera measures. Taxonomic abundance was normalized using bracket -R1 150 -IG based on the Kraken 2 output to estimate the abundance at the species level, the genus level and above. Decontamination and filtering of classified data. The normalized data from the bracket were combined according to the study design using the access VLOOKUP function. Then the combined normalized data were filtered and decontaminated using the decontam 1.0 R package using a prior-value threshold of 0.5. And these retain species that appear in more than 5 samples. The abundance data were filtered to remove low abundance species using a total abundance of 0.0005 as a threshold for all samples in each comparison for a downstream analysis.
Assalamualaikum warahmatullahi wabarakatuh. Today, I will presenting few element statistical analyzing and visualization on data. One of statistical analysis that can be measured is alpha diversity. Alpha diversity calculated based on ecological community with respect to its richness and evenness. Next, principal coordinate analysis used in dealing with numerical distance with no measurement error. This element calculated by Bray-Curtis. What is Bray-Curtis? Bray-Curtis means way to measure the dissimilarity between two different sites. Thus, other element that need be highlighted is constraint analysis of principal coordinated, also known as CAP. This statistical analysis allow a constraint ordination to be done on the basis of any distance or dissimilarity measure. Next, I will explain about resistomic analysis. Resistance means identify and quantify using short read. Thus, quantification on antibiotic resistant genes was performed using short break quantify option using resistomic marker. That's all from me. Thank you.
In this study, we found that the researchers have applied a molecular biology technique which is metagenomic shotgun sequencing, or we call metagenomic next generation sequencing (MNGS) rather than 16S rRNA gene applicant sequencing method. The reason why the researchers did not choose the 16S rRNA sequencing method is that even though this technique has been used widely, especially on the Lyme microbiota, according to previous studies, but still this technique has several limitations as this technique has potential deviation in microbial composition due to amplification bias. Also, it cannot detect most microorganisms at the species and the strain levels. Unlike shotgun sequencing, is unbiased because it captures all genomic DNA in a specimen rather than just one specific region of DNA, as in the application of the 16S rRNA sequencing method. Thus, shotgun sequencing can identify and profile more genera of bacteria, archaea, fungi, viruses, and various other microorganisms in samples that are useful for microbiome research. In addition, we have identified that this metagenomic shotgun will also be a more powerful approach and a suitable choice in studies to evaluate changes in the composition of the gut microbiome, in addition to detailed profiling antimicrobial resistant genes and their hosts.
The benefits of using this technique is able to identify the changing taxon at species level. It is beneficial to identify the changing taxon at species level as we can discover their function earlier. High relative abundance of species S. aureus significantly enriched in TB patients. S. aureus is a common pathogen that can infect multiple organs and cause critical disease, which may cause secondary infection in TB patients. Relative abundance of R. piquetii and P. melaninogenica were significantly lower in the untreated TB patients compared to a healthy patients. R. piquetii able to catalyze antienamide into active form against mycobacteria, while P. melaninogenica in untreated TB patients may weaken lung function and accelerate inflammation. Although difference in lung microbiota between healthy patients and TB patients were observed, it's been unable to determine whether the changes in taxon is a cause or consequences of tuberculosis infection. Next, it has unmatched comprehensive sequencing analysis of all microbial and host DNA. Metagenomic next gene sequencing captures all the sequence of all DNA or RNA present in a sample, unlike others, providing a high throughput technique to sequence not only entire microbiome but also human host genome. And lastly, it provides highly quantitative results with appropriate control. The quantitative result can be used to track the progressions of disease over time, evaluate the treatment success, and identify pollutants.
For the conclusion, first we have advantage and disadvantage. The advantage is MNGS is a biased sampling which enables the broad identification of pathogens in a hypothesis-free manner. For the disadvantage, that the majority of patient samples microbiome nucleic acids are dominated by human host background. After that, for the healthcare, the help that MNGS provide in healthcare is cut antimicrobial treatment versus conventional antimicrobial treatment in irreversible. Lastly, is MNGS are successfully used in this study with results that show pulmonary TB patient display unique lung microbiota signature distinct from that of healthy individual and lung cancer patients. The study also exhibit that anti-TB treatment increase alpha diversity and affects beta diversity in the lung microbiota. The data also indicated that the anti-TB treatment accelerates the enrichment of RGS.